welcome to the session Biomolecular Simulations, Structural Bioinformatics, and System Biology. Now we are going to start with Professor Mónica Pickholz from Argentina. She will be the first speaker of this session. And Dr. Mónica works in computational simulations of different problems of biological and pharmacological importance at the Physics Institute of Buenos Aires. She has more than 50 scientific articles related with biophysics and structural bioinformatics. And the title of her talk is going to be Polymeric Drug Nanocarriers Insights from Simulation. So Mónica, go ahead with your talk.
Okay, first of all, I would like to thank you very much for the invitation. It's a great pleasure in this situation to be here and share my work with all of you. I will talk and share the screen from different computers in some way to avoid noisy programs that I'm hearing. I don't know if you are seeing okay. I think
yes.
It
yes, is okay, but you have to share the full screen.
Yeah. I will put. It's okay like this?
Yeah, now it's okay.
Perfect, perfect. So today I will talk about polymeric drug nanocarriers and what kind of insights we can get from simulations. First of all, well, when we have a new pathology or a pathology, we want to find a drug or a treatment to treat this problem, no? But there is a very, very, very long road, and we have many, many drugs that are already approved for the FDA and MAT and the different agencies. When you have a drug, first we need to explore, develop, trying in vitro, in vivo, go to the different phases that we now all of the people know perfectly, the different phases that the drug should go through to be approved. But many of the drugs that are not as well as they expect, they have problems to reach the target. Then a different strategy could be develop nanocarriers, especially designed to improve some difficulties that could have the drugs. For instance, low solubility, or that they are severe adverse effects at the systemic level, or low acceptance for the patients or physicians, because they have to apply them very frequently and people forget it about. And then for each problem, we can think about a solution. Some have solutions and don't have. But our question is what we can help, how we can help from simulations. Well, then we are looking for a nanocarrier to improve the drug delivery to a specific drug. No? What is the ideal nanocarrier? The nanocarrier, as the name indicates, it's at the nanoscale particle. So the ideal nanoparticle should encapsulate the drug, a specific drug, take the drug to the pharmacological target, release the drug, and be completely removed from the organism. This is our, as a physicist, is our spherical, less neglectable mass curve. No, this is the ideal situation that we want to have a nanoparticle. Well, there is different strategies depending on which kind of drug do you want to encapsulate, where do you want to deliver, if they need to go through the blood system. For instance, you can add pegylate the nanoparticle to travel through. You can use natural lipids or different lipids. You can activate the nanoparticle with different external stimulation. You can use this also. I am talking about the general nanoparticle. You can use also as theranostic particle. Well, we have different strategies depending of the system. But okay, I will talk about some specific nanoparticles that we were involved with. First of all, most of the work that we carry out is using molecular dynamics simulations. So I will introduce, even if I think that all the audience know the technique, I will fastly introduce it. In molecular dynamic simulations, we solve Newton equations numerically. For that, we don't need to forget that atoms and molecules are quantum objects. But when in some conditions, we can treat as a classical system if we teach to the classical potentials how to interact and how to take into account many of the quantum properties. Well, with this, we solve Newton equations, and in some way we sample an statistical mechanic ensemble. Yes, we have different pictures of the system at different times. If we allow to do a long simulations, we can have many pictures and have too much statistics. And supposing that doing the ergodic hypothesis that say that ensemble average it's equivalent to temporal average, we can measure any property that we can write as a position or velocities of the particles that are involved or are part of the system. Well, this is a very short introduction. Because I am talking about the nanoscale, you can imagine that it's quite difficult to reach the nanoscale at the atomistic level. But we can gain some insight from the atomistic levels and go to other descriptions. What is the problem with the atomistic levels? The time scales that you can reach and the size of the system that you can study. So for many problems, you can alleviate these problems going to coarse-grained models. What is a coarse-grained model? Is you group in coarse-grained models, you group groups of atoms in a specific site or bit. So, for instance, if I want to describe a liposome that is a nanoparticle, if I do atomistic level, I will suppose that the liposome is it's very big, have a radius that tends to infinite. So I will take a planar lipid bilayer, and I can get many information from a specific interaction of the drug with this lipid bilayer. By the other hand, if I go to coarse-grained model, I can go to a very small but entire liposome. So I can get information of the entire system, but in a simplified way. So the reality will be in some way in between, but we can get information from both sides. It's important to have an interplay between the techniques to can get information and to can describe the systems. So I will start with one example. This example is about sumatriptan, that is an anti-migraine drug. This is an anti-migraine drug that was a drug from design, was a perfectly designed drug to be an agonist of serotonin. Yes? So they look at all the interactions to design this drug and look at the serotonin and improve the interaction with the serotonin receptor. But when they go to the practice, the drug was approved and is used and everything, but was not as good as they expect. What happened? That most of the receptors are inside of the brain, and then the drug should cross the blood-brain barrier. That is a bilayer somehow, and it's difficult for this drug to cross the bilayer. So we studied the interaction of this drug with the bilayer, changing the charge, structure, lipophilicity, concentration, initial conditions. The drug go to the interface and don't cross in any condition a simple bilayer. And they stop in the interface due to the different main interactions, like salt bridge, cation pi, hydrogen bonds, and etc. So the conclusion was, this is a good drug that needs a nanocarrier. Sumatriptan needs a nanocarrier to get in the right place. Well, our experimental collaborators suggest us to use poloxamers. Why? The pKa of this drug is around 9.8, and essentially it's a protonated drug, and it's most of the time it's charged, positively charged. So it's a very hydrophilic drug. So we need to look for a nanocarrier, a good nanocarrier for hydrophilic drug. Well, they propose a poloxamer. Poloxamer is a 3-block copolymer that have a center that is made from a hydrophobic monomer that is repeat M times, and is flanked by 2 tails of hydrophilic polymers. When they are in water, depending on temperature and concentration, they can assemble in a micelle, and depending on time, temperature, and concentration, they can form hydrogels. So, to do a long story short, we mapped our polymer in a coarse-grained model, yes? And for each monomer was replaced by a specific site in our coarse-grained model. We see the self-assembly of this copolymer in a micelle, and we also start with different pre-assembled micelles. So we are sure that our model form micelles in the size that we expect to find it. Well, but we want to encapsulate a specific drug. In particular, we are using or following the rules of the Martini coarse grain model. So in Martini coarse grain model, they have many lipids, many proteins, and some polymers, but they didn't have at this time the drug. So we need to build a coarse grain model for the drug. For that, we base our model, we map the 45 atoms of the drug in 7 specific sites, and we teach how to interact with the membranes. For that, we start with the atomistic, we compare the atomistic simulations that we already have done with the coarse grain coarse grain simulations with this new drug to fine-tune the parameters to have a good model, a good coarse grain model for the sumatriptan. Well, to do a long story short, we made it, we reproduced quite well the atomistic simulations with the POPC membrane, and then we go to the simulation of the whole micelle. We start from different initial conditions. First, or at the same time, we put all the drugs in water and all the drugs inside the micelle. And in all cases, we find that the drug partition very well in the hydrophilic tails of the micelle. So seems that at least for this kind of system, seems to be a very good encapsulator. There is many things that we should try, but this is an insight that we can give from this kind of simulations. So in this work, we see that sumatriptan loves interfaces, and then it's very difficult for it to reach the real target. For that, we propose encapsulate it in F127 micelles. F127, it's a specific poloxamer that have 65 units, hydrophobic units, and 100 in each flank of the tails of hydrophilic monomers. So we find get the encapsulation, we carry out the simulations and show that sumatriptan could be encapsulated in this kind of system, but we also went to the lab and observed the encapsulation and the change in the zeta potential that we have when they were absorbed at the surface or at the hydrophilic interface. Well, then our sumatriptan CG model captures the main behavior, and then we can be sure or assure that there is a good nanocarrier for the drug. In this problem, we have the drug and we want to look for a nanocarrier. Let me start another story. Continue with the polymers according with the title. There is very useful, or is very well known, liposomes as drug delivery systems, but the following this advice, we can do synthetic polymers that fit much better with our goals. Well, in this way, we choose a D-block copolymer that have hydrophobic part and hydrophilic part, following the idea of the lipid that have a hydrophobic head and hydrophilic tail. hydrophobic tails, yes. And then we put everything in water. We choose different sizes of these beads. We map the beads with this specific copolymer. We put everything mixed, water, polymer, and so on, and do the simulations until we reach a perfectly well ordered lipid bilayer, polymer bilayer. Yes, for this we consider a periodic boundary condition, NPT ensemble, but we see here in light blue is the water, in pink is the hydrophilic tails of the polymers, and in red is or orange is the hydrophobic. You see that we have a very hardcore of hydrophobic part of the polymers, hydrophilic that interact with water, and very well ordered bilayer. This bilayer represent in a polymerosomes the first approach that I talk about when we want to consider a big system and you just suppose that a radius is quite large, and you consider just a region, just a small part, and consider that it's a planar bilayer. Well, but I have said that you can tuning the relation between the hydrophobic and hydrophilic beads to have different size of the hydrophobic region or hydrophilic region or in interface, whatever, depending of the kind of drug that you want to encapsulate. So now we have the nanoparticle, but we want to look for a drug. We know that it's quite difficult to parameterize a new drug in a coarse grain, because we need to know a lot about the interaction to teach in a coarse grain model. But time ago, I was working a lot with local anesthetics, and we developed a coarse grain model for prilocaine. And prilocaine has a pKa of 7.8. So at this time, we study both the protonated and neutral forms of the molecule. So for this case, we have done, at this time, we have done the mapping, 36, 37 atoms, depending on the protonation state, and we map by 5 specific sites. We compare with the atomistic simulations, and we study the encapsulation into a liposome. So here we have a model, an interesting model, because we can test hydrophobic and hydrophilic drugs. When it's neutral, it's more hydrophobic and it goes to the center of the liposome. And when it's hydrophilic, when it's charged, we have similar results than with the other charged drug. So we want to see what happened with this model when we use neutral drugs. When we have neutral drugs, all the drugs go to the hydrophobic region and they stay there. I'm showing here the mass density profile that is going in this direction. If we go in this direction, they show the amount of mass that we have of different species. In blue, in light blue is water, pink is the hydrophilic tails, and orange the hydrophobic core. Well, this is the situation for the neat bilayer, and this is the situation for the bilayer when they have a hydrophobic drug. They go inside the bilayer, the core, the hydrophobic core of the bilayer. Well, what happened in the same kind of system when you put the hydrophilic drug? When you put the hydrophilic drug, we found a partition between the interface, a region of the interface and the water. And this partition, if we look at this number of this amount of drug in the interface, it increases when you increase the amount of drug. We can calculate a free energy of partitions between both phases, roughly estimated as minus RT, the logarithm, the natural logarithm of the difference, the relative amount in each, the proportion in each region. Well, within this model, so we find a coarse-grained model. We carry out to study the block copolymers and use a polymerisome as a model. We see the encapsulations in copolymer bilayers, both protonated and neutral prilocaine. We see that they encapsulate in different regions, and we went to very small polymerosomes, and we mix the pH, the physiological pH situation when you have a partition between neutral and protonate. I will not show here the results, but we find that we find both a kind of anesthetic partition in this, the entire liposome and also in the planar bilayer. And we investigate, we develop a tool to investigate the mechanical properties of this to see if we expect to be more stable or less stable with the incorporation of the drugs. Well, to finish this, talk, I will enter again a short talk about the same subject that Eliana Sciuto talked a few minutes ago, but from different perspectives. Other way to encapsulate drug is to link the drug to a nanocarrier. In this case, Eliana and all the collaborators want to link a mini-uno that Eliana and co-workers developed, and together with Eliana, we make long simulations to understand how the mini-uno affects the nanoparticle. Yes, I will not describe too much, but this is under review. This work is under review. But it's another insight that we can get from simulations in nanocarriers. And it's another way to deliver a drug for the nanocarriers. The whole paper is under review, but it's in the bioarchive if you want to read it. This is the title. Well, most of the work that I show here was done by Irene Good, Juan Albano, and Damián Grillo. And we have many collaborators in this specific work, especially Eneida de Paula, that opened her labs for us each time that we want. Rodrigo Portugal, that allows us to go to do cryo-TEM in the LA Nano in Brazil, in Campinas. Cristiano Oliveira, that once I show the electron density profile of the sumatriptan in a talk, and he said, I can measure these things that you can calculate. And I said, I have the drug. And 1 week later, we were in his lab doing the measurements of this system, and we can correlate the simulation with the experiments. Well, Marta Ferraro and Julio Facelli were involved in the polymer SOMS project. Marta was the director of Damián Grillo during his thesis. And well, I would like to thank again all of you for your patience and attention.
Okay, thank you, Monica, for your nice talk. And we have time only for one question now. So if someone has any question, you can ask or write it in the chat using the option questions and answer. Okay, I have just one question. Okay, so I'm curious if you use some specific software or server for parameterizing molecules in coarse grain.
In these cases, in our drugs, we didn't. We just compare with the experiment that we follow some rules that the Martini has, and then we compare with the atomistic simulations. Try to well describe the the results that we get from the atomistic simulations. You cannot do that in all situations, but in this case, we did.
Okay. Thank you, Monica, for your talk. And now...